everyone. So we're continuing on with the 2021 May paper 1 paper. So right now we're going to do question 5 and onwards. All right, so the diagram shows a sperm. Part A1, it says describe the function of a sperm. So the purpose of the sperm is to join with the ovum, which is the female egg, during fertilization. So we can say, oops, let me just draw that in purple. To swim to the ovum and join during fertilization. All right. Part 2. State 2 other structures apart from the mitochondria that can be seen in the sperm if it is viewed using an electron microscope. So an electron microscope has much greater resolution than a light microscope because you're using electrons that would be able to pick up a lot more detail. So I've just included an image of an electron microscope. This is a colored electron microscope. So if you recall, images from an electron microscope would be black and white, right? So they've just added color in this image, and this is from the textbook. So this is just to show you the kind of structures that you'll be able to see inside of the cell. So you can see mitochondria here that looks like this. You can see the rough endoplasmic reticulum. So the endoplasmic reticulum here it looks like it has little dots on it, and that's because of the ribosomes. That's why we refer to it as rough endoplasmic reticulum. And the function of the endoplasmic reticulum, or the rough endoplasmic reticulum, is for protein synthesis, and then it sort of helps package the proteins. Now we can also see the nucleus here, but even inside the nucleus as well, you can see some of the DNA that's sort of floating around, which is why in the marking scheme, you can also put down that you can see chromosomes as well. So let me just remove this. I'm just going to write ribosomes. So, whoops. Ribosomes, which are the site for protein synthesis, and then endoplasmic reticulum. All right, part B, explain why there are a large number of mitochondria found in the sperm. So mitochondria is where aerobic respiration takes place, and the function or the purpose of respiration is to produce ATP. And why is ATP relevant? Well, the sperm needs to swim, and so it gets that energy from ATP. So let's write that down. This is because aerobic respiration takes place in the mitochondria. Respiration produces ATP molecules which are needed for sperm to swim. All right, let's move on to part C. The actual length of the sperm between X and Y on the diagram is 3 micrometers. So it's referring to this distance here. This is the actual length. That's 3 micrometers. So you need to use your ruler for this and actually measure out the distance between X and Y. And so according to the marking scheme, the distance is 145 millimeters. Right, so the equation for magnification is magnification equals image size over actual size. The way I remember it is that it looks like MIA, M-I-A. So the image size here is 145 millimeters, and the actual size here is 3 micrometers. But before we do anything, we need to make sure that the units are the same for both of them, because one of them is in millimeters and the other one is in micrometers. So what we can do is convert, let's say, the micrometers here into millimeters. So 3 micrometer would give you, so here they've given you the conversion rate. So for 1,000 micrometers, that will give you 1 millimeter. So to get from micrometers to millimeters, we just need to divide that by 1,000. So 3 divided by 1,000, that would be 0.003 millimeters. So now we can plug in the numbers into the equation. So 145 minus 0.003. And if we write that down in the calculator, 145 divided by 0.003, that'll get you 48333. So 48333. All right, let's go on to question 6. All right, question 6, the diagram shows a Bowman's capsule and its associated blood supply. So this right here is the Bowman's capsule. Sometimes you'll hear it as the renal capsule. So renal is a term that we use to refer to the kidney. And then these capillaries that you see here, this is what we refer to as the glomerulus. So part 1, it says describe what is meant by the term ultrafiltration. So what happens during ultrafiltration, which is the first step of our kidneys filtering out our blood, is that you have this high blood pressure and that pushes out small molecules that are able to pass through from the glomerulus to the Bowman's capsule. So let's write that down. Okay, so this is when the high blood pressure forces out of glomerulus. Okay, so 1 mark for forcing out the small molecules, and the other part is under high blood pressure or under pressure. All right, part 3. State 3 differences between the composition of blood in vessel A and the composition of blood in vessel B. All right, so if we scroll back up, this is what we see. Blood vessel A has a much larger lumen than blood vessel B. So that's important to build high blood pressure. So I'm going to draw out a diagram here. So imagine if you had a hose or a pipe, right? And then it gets narrower towards the end. What would happen, so imagine the blood is flowing this way, what would happen is because it's getting narrower and narrower, it creates a lot of high blood pressure around here. It's sort of similar to, for example, on the road, if you have 5 lanes and then these 5 lanes sort of merge to become 2 lanes, you'll have a bottleneck, right? So it creates high blood pressure. So that means this is blood coming in and this is blood leaving. So in order to answer this question, we basically want to figure out what actually passes through from the glomerulus to the Bowman's capsule. And these are these smaller molecules. And these small molecules include glucose, includes amino acids, and includes urea. So we can actually write that down. So that means that blood vessel A, which is the blood coming in, has more glucose, more amino acids, and more urea. So blood vessel A has more glucose, blood vessel A has more amino acids. Blood vessel A has more urea. All right, let's move on to part 3. Explain how the blood vessel A and blood vessel B help the process of ultrafiltration. Okay, so I just mentioned that earlier. So I mentioned that because blood vessel B has a narrower lumen than blood vessel A, that creates high blood pressure, which then forces the small molecules out of the glomerulus and into the Bowman's capsule. So let's write that down. Okay, so we can say blood vessel A carries blood to the glomerulus because blood vessel B has a smaller lumen it causes the blood pressure in the glomerulus to increase. This in turn causes small molecules to be forced out of the glomerulus and into the Bowman's capsule. All right, let's go over the marks. So 1 mark for vessel A brings blood to the glomerulus. Second mark is that blood vessel B has a smaller lumen or a narrower lumen, and that causes the blood pressure to increase. That's the third mark, which then forces the small molecules to come out. All right, let's move on to part B. The diagram shows part of a kidney dialysis machine. Okay, so a dialysis machine is basically what patients that have kidney problems use in order to filter out their blood because if their kidneys aren't working as well to filter out the blood, what happens is that urea can build up in their blood and that wouldn't be too good for their body. All right, the dialysis fluid contains glucose at the same concentration as the blood flowing through the dialysis tubing. So basically what they're saying is that the amount of glucose molecules inside of the blood is the same as the concentration in the dialysis fluid. All right, suggest the effect of a person undergoing dialysis if the concentration of glucose was below that of the blood flowing through the dialysis tubing. So we know that diffusion is when molecules go from a region of high concentration to a region of low concentration. So if you have low levels of glucose in the dialysis fluid and you have high concentrations of glucose or higher concentrations of glucose in the blood, what would happen is that the glucose would diffuse from the blood to the dialysis fluid. And how would that affect the person? Well, if they have less glucose in their blood, then there is less respiration that's happening and therefore less energy. So let's write that down. So we can say glucose would diffuse out of the blood and into the dialysis fluid. This would mean less glucose for respiration, which would cause person to have less energy. So let's go over the marks. Number 1 is glucose would pass from the blood. The second mark is respiration would be affected, or respiration would be reduced. And the last one is lack of energy. All right, let's move on to question 7. A student uses this apparatus to investigate the energy content of different foods. So this is the student's method. Add water to the glass tube and then measure the temperature of the water, and then place a piece of food onto the mounted needle and set the food alight by placing it into a Bunsen flame. Then you'd place it under the glass tube and then measure the temperature of the water when the food stops burning. So the purpose of this experiment is to basically measure the energy content. So the higher the energy content of that food, the greater the temperature rise of that water. All right, repeat the method using different foods. Part A1 says state 3 safety precautions that a student should take. So let's scroll back up. So one obvious one is that they're dealing with burning food, right? So they want to be careful to avoid burns and maybe use a heat-proof mat, for example. Let me show you a picture of a heat-proof mat. So this here below the Bunsen burner is a heat-proof mat. So I'm going to remove that. Okay, so let's write that down. Another obvious one is you need to be using safety goggles. So the student should use safety goggles. All right. And another one is because you're using a mounted needle and it's very pointy, it's important to make sure that you don't poke yourself. All right, let's go on to part 2. State 3 factors that should be controlled during the investigation. So it helps to scroll back up to the experimental setup. So one of the first things that you need to control is the volume of water that's inside of the glass tube. And the reason why that's important is because a larger volume of water would require greater amounts of energy to increase its temperature. Another thing to keep constant is the mass of food that you've placed on this mounted needle. And that's important because if you have a larger mass of food, then that would have a greater energy content level, and that would affect the temperature rise of that water. And lastly, you want to make sure that the distance between the tube and the food is kept the same. That's important because the closer you put the food to the glass tube, the higher the temperature of that water would be. Right, so let's write this down. All right, so the volume of water and then the mass of the food on the mounted needle, and then lastly the distance between the food, I should say burning food, and the glass tube. Okay, part 3. Explain why the energy content of a sample of food is likely to be higher than the energy of the same sample measured by this method. Alright, so let's scroll up so we can see the experimental setup. So when it comes to calorimetry experiments, and if you guys do IGCSE chemistry, You'll also know this as well, but when it comes to this kind of experimental setup, there is always going to be heat loss. In this case, heat loss to the surroundings. There is also going to be heat that is absorbed by the glass tube here, and therefore not all of the heat is being transferred to the water, which is what we're measuring the temperature of. We can also say that because there's no apparatus to stir the water, the heat doesn't get distributed throughout the entire volume of water that we see here. So that could also lead to some inaccuracies. So let's write down, let's see how many marks this is. So for this, we're going to have to write down 5 different points. Okay, let's go over what I spoke about. So there is heat loss to the surroundings, and some of that heat could have been absorbed by the glass tube. Let's just scroll up. So it could be transferred to or absorbed by the glass tube here, or even the mounted needle as well. And the mounted needle. This would mean that not all of the heat is transferred to the water. So we can say another reason is that the water was not stirred, which meant the heat was not equally distributed throughout the water. So that's actually 5 marks. Another mark you can also say is perhaps not all of the food was burned to release that energy. Let's go over the marks. So we have heat is lost to the surroundings, or heat is lost to the atmosphere. We also have heat could have been absorbed by the glass tube or the mounted needle. And then the third mark would be not all of the heat is transferred to the water. That's 3 marks so far. Another mark is water was not stirred, or there's lack of stirring, and that led to an uneven distribution of the heat throughout the water. All right, let's move on to the next question. All right, let's scroll down. Okay, the diagram shows a calorimeter. This is a piece of apparatus that is used to measure the energy content of a sample of food accurately. So here we have the food that's burning here. We've got water that is surrounding this vessel or this container. And then we have a thermometer here to measure the temperature of the water. So here it says, discuss the features of this calorimeter that will give a more accurate measurement of the energy in a sample of food than the method used by the student. So one thing that I first noticed is that there is a lid, and you can see that there, and that prevents heat from being lost. That's one thing. Another thing is that, do you see where it says the heat transfer coil? So that part is used to transfer the heat from this burning food to the water, and it does it more efficiently as well. Another thing that we notice here is that it comes with a stir, and that ensures that the heat is distributed throughout the entire volume of water. And then lastly, we have oxygen supply. And the reason why that's important is because it ensures for complete combustion. And when complete combustion occurs, it allows for all of the energy in that food to be released. All right, let's write that down. So there is a lid to ensure that less heat is lost to the surroundings. So there is a heat transfer coil to make sure the heat is transferred to the water. All right, there is a stir to distribute the heat in the water. Lastly, oxygen is supplied to make sure complete combustion occurs. All right, let's go over the marks. So we can say there is a lid to ensure that less heat is lost or less heat escapes. There is a heat transfer coil to make sure that the heat gets transferred to the water. There is a stir to distribute the heat in the water. And then we have oxygen is supplied or oxygen is used to make sure complete combustion occurs. So that's 1, 2, 3, 4, 5 marks. All right, let's move on to the last question of this paper. Question 8. The diagram shows a reflex arc involved in a reflex action. So part 8 says describe the function of a reflex arc. So we got to mention what it is. So, wrong colored pen. All right. A reflex arc is an automatic rapid response to stimuli. So that's actually 2 marks, one for mentioning automatic and the other one is rapid response. And then what is it used for? What is the reflex action used for? This occurs to protect the body from harm. And that would be the 3rd mark is protect. Now, if you want to add a 4th mark, you can also give an example. Oh, actually, it says from harmful effects. So protect from harm. That would actually be 4th mark, sorry. All right, let's move on to part B. Using a line labeled Z, show on the diagram which part of the reflex arc would be connected to the receptor. Okay, so your receptor detects the stimuli, so that could be receptors in your skin, receptors in your tongue, for example. And then what goes after the receptor is the sensory neuron. and that sensory neuron has electrical impulses traveling from the receptor to the relay neuron or the interneuron, which is what we see over here. So if this is the, let me write this down, if this is the sensory neuron, then this, how do we label it again? Label it Z. This would be Z. Okay. Part C. Describe how the nerve impulse passes from position X to position Y in the reflex arc. So let's look at position X and position Y. So this is X over here, this is Y over here, and between them we have a synapse. So let me just include a diagram of what a synapse looks like. All right, so here I've included a diagram of the synapse. So what happens here is you've got an electrical impulse that travels down the axon and finally arrives at the synapse. And these causes the neurotransmitters that are inside of the vesicles to release the neurotransmitter into this part here, which we call the synaptic cleft. And then when it gets released to the synaptic cleft, it would diffuse across and bind to receptors that we see in the postsynaptic neuron. And then afterwards, the electrical impulses get triggered onto the postsynaptic neuron. All right, let's write that down. This is a 6-mark question, so there's a lot to write down. So first, the electrical impulses arrive at the synapse, which causes the vesicles containing neurotransmitters to be released into the synaptic cleft. The neurotransmitters, you can give an example of neurotransmitters like acetylcholine. That would actually give you a mark, but anyways, let's continue. So the neurotransmitters diffuse and bind to receptors found on the postsynaptic neuron. This triggers electrical impulses to travel down that postsynaptic neuron. Oops. Let's just write neuron. Okay, let's go through the marks. All right, so here we have synapse here, and then it causes the release of neurotransmitters. So it causes the neurotransmitters to be released. That's the second mark. The third mark is from the vesicles, right? That's a third mark. And then it says diffuse across the synaptic cleft. We kind of mentioned that there. That's the fourth mark. Fifth mark is bind to receptors that are found in the postsynaptic neuron. And sixth mark is trigger the electrical impulses. All right, so that's the end of paper 1, May 2021.